So proceeding forward, in this segment, we have two excellent speakers, and they will have a theme of diseases. The first will be focused on angiosarcomas, and the second will be on intimal sarcomas, reflecting the disease subtype that we commonly see in cardiac sarcomas. So Dr. Paul Huang from ICR in London is going to talk about angiosarcomas from a bed-to-bedside approach. Dr. Huang is the head of Molecular and Systems Oncology Laboratory at the ICR in London. He holds a PhD in biological engineering from MIT in 2008, and he's currently also a reader in molecular and translational oncology at ICR. His lab focuses on understanding aberrant signal networks and drug resistance in sarcomas with the goal of developing biomarkers and new therapies in rare diseases, including in cardiac sarcoma. Paul is also the Deputy Director of the Joint Royal Marsden/ICR Sarcoma Research Center, and he serves on a number of committees to include the Vice Chair of the Pathology and Translational Research Committee at the EORTC Sarcoma Group, and he was also elected as a Fellow of the Royal Society of Biology in 2020. Without further ado, we will proceed with the presentation by Dr. Huang. Thank you.
I'm Paul Huang, a research group leader in the Division of Molecular Pathology at Institute of Cancer Research in London. I'd like to thank the organizers for the kind invitation to speak at this fantastic cardiac tumor conference. And the topic of my talk today is angiosarcomas, a bench-to-bedside approach. Angiosarcomas, as many of you are aware, soft tissue sarcomas are endothelial cell origin. And they are fairly rare in that they comprise just 1 to 3% of soft tissue sarcoma subtypes. But is in the context of today's talk, cardiac sarcomas represent about 30 to 40% of these tumors. They have a poorer prognosis compared to other soft tissue sarcoma subtypes. So here is a series of just under 1,800 patients, and you can see that angiosarcomas have inferior overall survival compared to other soft tissue sarcomas. And there is a huge amount of clinical heterogeneity. So, for instance, there is heterogeneity in terms of etiology. So primary and secondary angiosarcomas, secondary angiosarcomas include radiation-associated angiosarcomas as well as lymphedema-associated angiosarcomas. And there are a range of different anatomical sites. This is obviously not exhaustive, but here it just lists some of the key sites for which angiosarcomas have been found. So for the purposes of today's talk, I'm going to focus on three main areas. The first is on molecular profiling studies and what we have learned so far. The second is the characterization of the immune landscape of angiosarcomas. And finally, really going back to the title of this talk, bench to bedside, how can biomarkers be used for clinical management of angiosarcoma patients? So focusing first on molecular profiling studies. So the current state of art for tissue omics profiling is really outlined in this slide. Here, there are a range of different methodologies and different things you can measure. They include mRNA transcripts, DNA copy number aberrations, DNA methylation, mutational level, whether they be exons or genomes, microRNAs, as well as proteins, and the list goes on. By far, the most comprehensive tool that's been used to study angiosarcoma so far has been primarily DNA sequencing, in particular next generation sequencing outlined here. There are many different methodologies that have been used. These include targeted sequencing panels, which I think many of you in the clinical community are very familiar with, where you look at specifically subset of genes, potentially cancer-causing genes that are of interest. There's whole exome sequencing, which sequences all the exons of the of known genes, which is 1 to 2% of all human DNA. And then there is whole genome sequencing, where you basically sequence everything, but it's bioinformatically quite intense in terms of data analysis. And all these different types of sequencing have their pros and cons, whether it be cost, coverage, as well as ease of analysis. Over the past 10 or 15 years, there have been a number of recurrent genomic alterations identifying angiosarcoma. I've just summarized some of the key highlights here, if you will. One of the earliest, and I guess most studied, was MYC amplification in angiosarcomas, which are enriched in primarily secondary angiosarcomas, although there are some primary angiosarcoma cases that do display MYC amplification. TP53 mutations, loss of function, have also been identified to be quite common in angiosarcomas, and they've been shown to be a driver, so driving angiosarcoma development in both mouse and zebrafish models. And unlike many of the other putative genomic aberrations identified in angiosarcoma, this remains one of the few that have actually been functionally shown to be a driver for this disease, at least in animal models. There has also been mutations and amplifications of angiogenesis signaling pathway genes, for example, KDR receptor, and also other signaling pathways. And there's a very long list here. I've just highlighted a few: PIK3CA mutations, DNA damage repair genes, ARID1A, which is a member of the SWI/SNP chromatin remodeling complex, ATRX, which is important for alternative lengthening of telomeres, NOTCH, as well as CHIP rearrangements and mutations. Over the past recent years, probably 5 years or so, really led by a Broad Institute, Corey Painter at the Broad Institute, this project, the Angiosarcoma project, was conceived really as a patient-partner collaborative science. It'd be remiss of me not to mention this collaboration when talking about angiosarcoma genomics. So what is the Angiosarcoma project? This is a snapshot of its website. It's really an effort to, across the US, collect not just patient tissue, but also patient medical information, as well as patient-reported outcomes, to develop a comprehensive resource that can drive discoveries for angiosarcoma. So again, this is taken from their website in terms of how they participate. You basically fill out a simple online form. You give permission to collect samples and data. And then these tumors, for instance, are then going for sequencing analysis. This was the first paper that was published by the Angiosarcoma Project 3 years ago now. And you can see here the coverage of the samples and data that were collected across the US, just to give you an indication of how a patient-partnered approach can lead to really collection of a large amount of information for a particularly rare cancer. And in terms of what was actually collected, you can see that number of patients registered at that point in time when the paper was published was about just over 300 cases. Contrasting that, of course, we may not be able to collect tissue specimens from all patients, but about 100, just under 100 patients tissue was collected for. And when this tissue was sequenced at the DNA level, the authors found something quite interesting, which was that a subset of the head, neck, face, scalp cases had an enrichment in signature 7, which is the UV-associated mutational signature. In addition, further analysis done by a second team here, published in Genome Medicine, found that in these cases from the Angiosarcoma Project, they had a tumor mutational burden which was much higher than the average across all tumor types in the Cancer Genome Atlas Consortium on multiple cancer types, and many of them actually higher than the average in melanoma, which is known to have a high tumor mutational burden. So this was a new finding, which will have clinical implications, which I will mention towards the end of my talk, that head and neck, face, scalp angiosarcomas have high tumor mutational burden and a characteristic UV signature. This has now been validated in multiple groups across a range of different anatomical sites for angiosarcoma. On the left here is data shown by the group of John Trent. And you can see here that when they looked at the tumor mutation burden across a range of different anatomical sites, you can see here that head and neck have the highest frequency in terms of tumor mutational burden, whereas visceral tumors have relatively low tumor mutational burden. The data on the right is from the Dutch group, shown here in the second reference, and you can see here again that the what they term to be UV associated, which is largely the head and neck face scalp patients, have much higher tumor mutational burden compared to other angiosarcomas, including the heart angiosarcoma shown in this reddish color on the left here. So this really confirms that at least at the genomic level, tumors from different anatomical sites, even though they're all angiosarcomas, have different genomic characteristics, of which tumor mutational burden is one of them. In the context of cardiac angiosarcoma, a paper published in 2005 found this very interesting mutation in POT1 in families with nephromoney like but TP3 mutant, TP3 negative families. When they sequenced lymphocytes and lymphoblasts from these patients, what they found is that there were POT1 mutations in these families. So here are 3 family trees shown in this diagram here. Anything in these red circles or squares indicates angiosarcoma patients. So you can see in this family 1, there were 3 angiosarcoma, cardiac angiosarcoma cases. And in family 2, there were 2 cardiac angiosarcoma cases. And in family 3, there was 1 angiosarcoma, cardiac angiosarcoma case. And so what they did was to sequence these patients, and what they found was that these patients actually had a recurrent mutation, the R117 missense variant, which was found in families which had cardiac angiosarcomas, but not in families that didn't have cardiac angiosarcomas. So what is POT1? POT1 is a protein in the sheltering complex, and this complex is important for regulation of telomerase activity, which is important for maintaining telomere length in normal cells. And what they found was that if you look at cells from patients which have the R117C carrier, POT1 levels, because of these mutations, actually lose expression of this protein compared to wild-type patients shown here. And this is in family 2 and family 3. In addition, they found that there was reduced telomere-bound POT1 using a second marker, a red marker for a telomere protein, TRF1. You can see that in the carriers for the R117C, you get a reduction in telomere localization of POT1 compared to those which are wild-type. And finally, if you look at the lymphoblasts from POT1 wild-type patients versus R117C carriers, you find that there is a slight but not statistically significant increase in telomere length. But what they managed to get their hands on was actually the cardiac angiosarcoma patient material itself and the tumor. And when they sequenced the tumor, when they evaluated the telomere lengths on these patients, they found that these tumors were significantly increased in telomere length compared to wild-type patients, really highlighting the importance of this mutation in terms of controlling telomere and may be causative for driving familial cardiac angiosarcomas. This has since been found POT1 to be a mutation in other forms of sarcomas, angiosarcomas, in a small percentage of patients. The next thing I want to move to is this idea of molecular heterogeneity, and this has been evaluated using a number of different omic approaches. So, for example, using transcriptomic approaches, whether they be targeted transcriptomics using a specific pathway panels, or global transcriptomics, whether it be RNA-seq or gene expression arrays. Multiple groups have now shown that if you just take transcriptomic data and do unsupervised clustering, what you're able to show is that these patients actually fall into different groups. So for example, this was the first study done by Christina Antonescu using gene expression arrays, microarrays, and she showed that, shown on the left here, patients that were had secondary breast angiosarcoma separated quite nicely from those patients with primary breast or primary soft tissue angiosarcomas. Similarly here, in this study, using a targeted panel from NanoString, a group from Singapore led by Jason Chan showed that if you take this gene expression data and you did unsupervised clustering based on signaling pathways, you can see that there is a group of angiosarcoma patients who had very low levels of all these signaling proteins. One group of patients with high levels of particularly proliferation proteins, proliferation pathways, Wnt segment, MAP kinase, PI3 kinase, and another pathway, which is another cluster which has upregulation of immune signaling proteins. So this really tells us very clearly that angiosarcoma is not one disease at a molecular level, and there are different groups of patients that fall into different categories. In a similar vein, but using a different methodology now, instead using methylation analysis, a group from a collaboration between the Germans and the Dutch looked at a panel of a cohort of angiosarcoma patients, and what they were able to find was two main groups when you clustered unsupervised clustering of the methylation data. On the left here, cluster A is primarily made up of what they term the UV-associated angiosarcoma. This is primarily, again, the head, face, neck, scalp cases, and the radiotherapy-associated angiosarcoma. On the right, group B here is mainly soft tissue as well as visceral angiosarcomas, although there are a subset of UV-associated and radiation-associated angiosarcomas. Now, if they looked at the, by sequencing the levels of chromosomal aberrations in these cases, they found that cluster A cases on the left here significantly higher levels of chromosomal aberrations compared to cluster B. And when they look at survival outcome, you can see here that cluster A cases on the left have better overall survival compared to cluster B cases. So focusing again on the methylation patterns, this suggests to us that different angiosarcomas have different molecular profiles, although the biology of these methylation marks may not be particularly well studied at this point. And finally, our group has done proteomic analysis, the subset of 30 cases from the Royal Marsden. And when we've done unsupervised clustering of the proteomic data, we find three different groups of patients shown here. In red is high protein expression, in blue is low protein expression. You can see there are different protein profiles. There's not real clustering between the primary and secondary cases, as well as anatomical site, suggesting to us that potentially this is separating cases based on biology rather than any particular clinical characteristic. That being said, we can actually undertake supervised analysis to identify the genes that, for example, separate the primary and secondary cases. So that's what we show here. You can see quite clearly that in the in red here, these are the genes that are upregulated in the primary cases, and on the left here, these are the genes that are upregulated in the secondary cases. And when we do ontology analysis to see what genes are what pathways are upregulated in the secondary cases shown here, you can see that MYC targets are enriched, which is consistent with what I told you just now, that MYC amplification is primarily found in secondary angiosarcoma. So this tells us that at least downstream at the pathway level indicated by proteins, this is also the case that MYC pathways are activated. So just to summarize the molecular profiling section of this talk, several actionable genomic alterations have been identified. I do want to stress that many of these are candidate drivers of the disease. The majority of them has not been functionally shown to be drivers, whether they be in animal models or in preclinical cell line models. Higher tumor mutational burden and a UV signature has been found in a subset of phase and scalp angiosarcomas, and this actually has been validated now by multiple groups. The POD1 mutation is important in familial cardiac angiosarcomas. And there is substantial molecular heterogeneity. Angiosarcoma is not one disease. And at this point in time, all the molecular clusters that I've shown you so far is really nice for understanding the biology, but unfortunately at this point, there's no clinical utility yet. But I'm sure we will find clinical associations and, for example, new targets in the future. So moving on to the second part of the talk, which is characterization of the immune landscape. As many of you know, the immune microenvironment is an important component in many different cancer types and in sarcoma as well. Here is just a diagram showing you the different types of cells which are found in the immune microenvironment, including lymphocytes here, macrophages, as well as dendritic cells. But what's key is that many of the drugs that we currently use, or and also novel agents in development, affect the immune microenvironment in many ways, whether it be a standard chemotherapy, which has been shown, for example, to lead to immunogenic cell death and upregulation of the MHC class 1 molecules, or anti-angiogenics, such as some of these tyrosine kinase inhibitors, which have been shown to modulate levels of different immune cells, as well as immune checkpoint expression levels, and a range of other novel immunotherapeutic modalities highlighted here as well. Now, there are a number of different ways to measure the immune landscape, two major ways, one of which I've shown here, which is basically just looking down the microscope and counting the number of cells. There are now multiplex immunofluorescence approaches. I've shown you one example here, which is using a black vector system where you can look at 6 different colors at one time. There are other more sophisticated methods like the Codex system, where you can look at up to 80 or 90 different colors at one time. But this just gives you the pipeline of how it actually deconvolutes the data and does automatic identification of cells and cell counting. In this particular example, we look at 5 different markers, including CD4, CD8, FOXP3 for regulatory T cells, natural killer cells, as well as macrophages. So using these type of approaches, scientists have gone into actually looked at the immune landscape of angiosarcoma. And here's one example here by the Dutch group reported in this study last year, where they looked at using the vector system to measure the levels of 5 different immune cells, and they've split it up by primary and secondary angiosarcomas, as well as different either etiology or anatomical signs. I just want to highlight here the cardiac angiosarcomas are shown here. Now, this figure is really hard to read, so I've taken the table out, which is much clearer. And what you can see here is that the UV, what they define as the UV-associated secondary angiosarcomas have the highest levels of immune cells across the board, whether they be different types of T cells or B cells. And along those lines, those the heart angiosarcomas have some of the lowest immune cell counts out there, suggesting that cardiac angiosarcomas are actually pretty immune cold. Although there is this one case which has relatively intermediate levels of immune cells. So again, heterogeneity even within a single anatomical site. Another way of looking at the immune landscape is actually using immune cell deconvolution technique. So rather than actually looking down the microscope for specific immune cells, you take profiling data like transcriptomic data, and you actually deconvolute the bulk transcriptomic data using, for example, single cell markers, cell type markers, to actually try to estimate the different immune cells that are within your sample based on a bulk RNA level measurement. And using this approach, John Tran's team has actually looked across a range of different tumor types, and they deconvoluted, a range of different angiosarcomas from different primary tumor sites, and they use deconvolution approaches to look at not just immune cell populations and stromal cell populations, but also various immune checkpoint genes. And what they found was there were 4 different immune classes. One in particular was termed the immune high cluster, shown on the left here, cluster 1. You can see it's pretty much red by everything indicating very high levels of not just immune cells, but also checkpoint genes. On the other end of the spectrum, we can see that what they've termed the immune desert group of patients, where basically everything is pretty low. So they are very immune cold tumors. Again, I will talk a little bit about the implications for this for immunotherapies in the third section of my talk. So just to summarize the immune landscape component of this talk, there is clearly heterogeneity in the immune landscape of angiosarcoma. There is a spectrum from hot to cold tumors. Secondary angiosarcomas generally have more immune cell infiltrates than primary angiosarcomas. And the analysis of a limited handful of cardiac angiosarcoma cases suggests that it's mostly a cold tumor, although there are exceptions. But the thing really to highlight here is these are all observations. A lot more work needs to be done in understanding the immunobiology of angiosarcomas. Now moving on to the third section of my talk, bench to bedside biomarkers for clinical trials and management. So what really are the barriers to implementing precision medicine in sarcomas? I've highlighted 2 here. Of course, there's a very long list, but these are 2 which I think are important. The first is we have a very poor understanding of sarcoma biology and heterogeneity, and I've already alluded to this in my previous 2 sections. And secondly, we have an incomplete knowledge of the factors that drive patient relapse and or drug response and resistance. And if we can understand, have a better grasp of these 2 aspects, the hope is that we can identify biomarkers for personalized therapy and use these biomarkers to design the next generation of smart clinical trials. So what are biomarkers? Really, biomarkers are an exercise in reducing heterogeneity. So if you have a mixed population, then we actually stratify them by whatever factor you wish to use, which may give us an indication, for example, whether therapy is going to work or not. Now, why is this important? Well, just an example, a very recent example here, a phase 3 study, the TAPAS trial, which is TRC 105. This is an antibody against endoglin and pazopanib versus pazopanib alone in patients with advanced angiosarcomas. And you can see here that although the trial was a negative trial, in the combination arm B in red here, there was a subset of patients, about 10% of patients, for which there was durable, but relatively long durable patient benefit. The question really is, how do we a priori identify these 10% of patients? And this is what I'm going to talk about. And really the question is, with all the heterogeneity that I've talked about today in angiosarcomas, is that even possible? We have heterogeneity at histological level. We have heterogeneity in terms of age of patients. We have anatomical site heterogeneity. I've talked just now about molecular heterogeneity and also immune landscape heterogeneity. So is it even possible to stratify these patients? So I'm going to give you 2 examples. The first example is really, I would say, probably the most promising for angiosarcomas, which is biomarkers for immune checkpoint inhibitors and immune checkpoint blockade. Now, in other cancer types, for example, melanoma, a number of candidate biomarkers for immune checkpoint blockade response have been identified. These include high tumor mutational burden, high expression levels of immune checkpoint proteins, general immune hot, for example, increased tumor immune cell infiltrates. And other studies in soft tissue sarcomas have identified potential biomarkers such as the presence of tertiary lymphoid structures and B cell signature. And really the first study I would say that made this link, although there was a number of anecdotal and case study reports of angiosarcoma patients responding to immune checkpoint blockade, the one study that really made the potential link between the 2 was the Angiosarcoma project. And I just want to, as a reminder again, that these patients have very high tumor mutational burden and a characteristic EUV signature. In fact, one of these patients was treated with with immune checkpoint inhibitor and actually had a long-term durable response. So at least a subset of patients of these head and neck face scalp cases had long-term responses to immune checkpoint blockade. And so following on from that, there have been a flurry of studies that have been published looking at this aspect. One of them is this study here, which was published in 2001, looking at 35 angiosarcoma patients, where they undertook genomic and transcriptomic analysis, and they correlated it with response to immune checkpoint blockade-based therapy. And the main take-home messages are as follows. No single traditional biomarker, whether it be TMB, PD-1, PD-L1, TILs, or the UV mutational signature was associated with PFS. So at least in the context of angiosarcoma, none of these can be used, or at least are necessary for a response to checkpoint blockade. Now, when they looked at a smaller subset of patients looking at transcriptomics and then using again deconvolution methods, they found that patients which were responders defined as greater than a 16-week response had higher levels of specific immune cells, and these are cytotoxic T cells, dendritic cells, as well as NK cells shown here. However, non-responders, shown on the left here, had higher levels of cancer-associated fibroblasts, and this just gives you an indication also that these are on those that did respond have generally higher levels or are more immune hot because they also express higher levels of checkpoint proteins like CD274, which is PD-L1, LAG3, and CTLA4. So, of course, this is still exploratory and needs to be validated, but I suggest that potentially looking at some of these signatures, whether by deconvolution or by IC, may be useful. A more recent pilot study looking at a combination of IL-2 agonists in combination with a immune checkpoint inhibitor. Here was a cohort of different sarcoma patients, of which there were 8 angiosarcoma patients. Interestingly, although the overall response rate was the highest in angiosarcoma compared to all the other sarcoma subtypes in the study, all the responders were actually non-cutaneous, and this is, I guess, opposite from what you would expect from the studies coming out on the head and neck face scalp cases. And in particular, on treatment, but not baseline, CD8 T cell levels as well as PD-1 associated overall response rate. Now, again, using transcriptomics, looking at baseline as well as on treatment, again, using the immune deconvolution methods, they found that patients which had higher baseline as well as on treatment tumors had partial responses, and these were the largely, at least defined by transcriptomic immune hot tumors. Interestingly, again, exploratory and needs to be validated, the Hedgehog pathway identified by transcriptomic gene set enrichment analysis was associated to resistance to this class of drugs. So this is all very interesting, and really shows you the vibrant nature of future research in this field. And I'm confident that we'll be able to find biomarkers for checkpoint blockade, immune checkpoint blockade for angiosarcomas in the very near future. The second class of drugs I want to focus on is multi-target kinase inhibitors, aka anti-angiogenics. And many of you are familiar with this class of drugs that are used, for example, pazopanib in sarcoma. Now, tyrosine kinase inhibitors as a class are, I would say, the poster child for biomarkers, because there are many tyrosine kinase inhibitors for which there are clear biomarkers used before you can give the drug to the patient. So, for example, EGFR inhibitors in non-small cell lung cancer, you need to have a mutation in particular mutations in EGFR. MEK inhibitor in melanoma, you need to have a mutation in B600E. Now, all the anti-angiogenics that are currently used in the clinic, whether they be pazopanib in soft tissue sarcoma, sorafenib in HCC, sunitinib in kidney cancer, none of them have any biomarkers for when we give these drugs to the patients. And perhaps it's not surprising that many of these benefit from many of these drugs, I would say, are relatively poorer compared to some of these specific TKIs and EGFR inhibitors or the MEK inhibitors. So why is it been challenging to identify biomarkers for these drugs? These are my view. There are a range of different cancers which are driven by single drivers, sarcomas with the exception of cancer types like GIST. Sarcomas probably are the class of cancers which are not driven by single drivers, so it will be difficult potentially to assign a particular mutation or biomarker for tyrosine kinase inhibitor. Secondly, many cancers are driven by multiple complex processes, not just tumor intrinsic, not just the tumor cells, but also tumor extrinsic, the tumor microenvironment, the immune cells, the stromal cells. All of this, the tyrosine kinase inhibitors have an impact on these cells as well. So it's not just tumor cells, but it's also the microenvironment. And finally, these drugs, particularly the anti-angiogenic agents, although we call them anti-angiogenics, they hit a range of different things, both in terms of the endothelial cells, but also in the tumor cells. And this is termed as polypharmacology. They do not just hit the EGFR or the BRAF or the ALK, they hit a range of different cellular targets. And so really, I would say for this class of drugs, we need to have a new ways of thinking about biomarkers for stratification, not just one gene, one drug, but really, or one mutation, one drug, but really perhaps a combination of different things. And so this was what was done recently in this study that was published in the European Journal of Cancer very recently, where they looked at a large cohort of sarcoma patients in the German DKTK consortium, which were given pazopanib. And you can see here they identified a 3-gene signature which comprised of gene expression levels of 3 receptor tyrosine kinases, the NTRK3 gene, IGF-1R low, and KDR high. And they call this the pazopanib predictor. And what you can see here is that if you are class in the PEP or the predictor low group shown in red here, you have very poor PFS when you're treated with pazopanib, whereas you're in the PEP 3 group, you have much better outcomes. On the left here is a training data cohort, and the right here the validation cohort. So obviously this needs to be prospectively validated. In each of these studies, there was less than a handful of angiosarcoma cases. So it's very difficult to say whether this will be broadly applicable to angiosarcoma, but it gives us a starting point in terms of looking to see whether this 3-gene signature will be useful in angiosarcoma patients and will need to be prospectively validated. So just to summarize my talk, I hope to have impressed upon you today that angiosarcoma is a molecularly heterogeneous disease, and there are a lot of new technologies coming out, such as single-cell sequencing and spatial sequencing that can provide new biological insights to address this issue of heterogeneity. Immune checkpoint blockade is important, but biomarkers for responders need further investigation. Understanding of molecular heterogeneity and immunobiology is really still at its infancy. And as we study a little bit more about the biology of angiosarcomas in the next 5 or 10 years, I think this will be key in terms of defining new therapeutic targets and biomarkers for this group of heterogeneous diseases. And finally, international collaborations, and I think this audience already knows this, will be key, in particular for rare tumors such as cardiac angiosarcomas. And with that, I thank you for your attention, and I'm happy to take any questions.